Good day, everyone. The next topic I'm going to introduce to you will be the Marcelo-Fernan Bridge of the body. My name is Dr. Kline Lim, and welcome to the Neuromuscular Junction. Yes, I said bridge. While the Marcelo-Fernan Bridge connects Cebu and Lapu-Lapu City, the Neuromuscular Junction acts the same way, where it serves to connect the nerve and the muscle. The body of water functions as a synaptic cleft, which serves as a space between the muscle and the nerve. Now, before we delve into the specifics of the neuromuscular junction, it is important to know that there are two types of synapses, chemical and electrical. Chemical synapses are brought about by action potentials which causes release of neurotransmitter substances from a presynaptic neuron. But this takes place longer to occur, which causes delay, and this is termed as synaptic delay. A good example of the chemical synapse is our topic for today, the neuromuscular junction. Electrical synapse, on the other hand, is brought about by two excitable cells that communicate by direct passage of an electrical current between them. The nerves in your body are examples of electrotonous transmission, which refers to the passive spread of charge inside a neuron and between cardiac muscle cells or smooth muscle cells. While gap junctions are specialized intracellular connections between a multitude of animal cell types. They directly connect to the cytoplasm of two cells, which allows various molecules, ions, and electrical impulses to directly pass through a regulated gate between cells. Now remember that the neuromuscular junction is also known as the myoneural junction or motor end plate, and is actually an excellent model of a chemical synapse. A neuromuscular junction is a chemical synapse between a motor neuron and a muscle fiber. It allows the motor neuron to transmit a signal to the muscle fiber, causing muscle contraction. Muscles require innervation to function and even just to maintain muscle tone, avoiding atrophy.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber.
So just a brief summary. Skeletal muscles are innervated by large myelinated nerve fibers that originate from large motor neurons in the anterior horns of the spinal cord. And take note that each nerve ending makes a junction with the muscle fiber near its midpoint. So let me introduce to you the parts of the bridge. The features of the neuromuscular junction through explaining the physiologic anatomy of the motor end plate. Picture A shows you the longitudinal section through the end plate, and picture B shows you a surface of an end plate. So the nerve fibers form a complex of branching nerve terminals that invaginate into the surface of the muscle fiber but lie outside the muscle fiber plasma membrane. The entire structure is called the motor end plate. It is covered by one or more Schwann cells that insulate it from the surrounding fluids. Alright, so here's physiologic anatomy number 1. As a motor nerve approaches the neuromuscular junction, it loses its myelin sheath and divides into fine terminal branches. The terminal branches invaginate into the muscle fiber at the center of the plasma membrane. Here. Terminal branches are covered or insulated by one or more Schwann cells, which serve to insulate it from the surrounding fluid. And the synaptic gutter, this is an invaginated membrane, and this is the point of a terminal branch which invaginates into the motor end plate. This is also known as the synaptic throat. The space between the terminal branch and the muscle fiber membrane is called the synaptic space or synaptic cleft. This space is 20 to 30 nanometers wide, and it is bound on one side by presynaptic terminal here and have a postsynaptic terminal on the other side. The presynaptic neuron or terminal is always an axon terminal. At the bottom of the gutter are numerous smaller folds of the muscle membrane called subneural clefts, which greatly increase the surface area at which the synaptic transmitter can act. In the axon terminal are many mitochondria. So do you remember the powerhouse of the cell? So this one supplies adenosine triphosphate or ATP, the energy source that is used for synthesis of an excitatory transmitter called acetylcholine, which is the primary neurotransmitter used in the neuromuscular junction. So we'll talk about it later on. The acetylcholine in turn excites the muscle fiber membranes, and the acetylcholine is synthesized in the cytoplasm of a terminal. But it's absorbed rapidly into many small synaptic vesicles, about 300,000 of which are normally in the terminals of a single end plate. In the synaptic space are large quantities of the enzyme called acetylcholinesterase, which destroys acetylcholine a few milliseconds after it has been released from the synaptic vesicles. Alright, so we'll discuss about the secretion of acetylcholine by the nerve terminal. Acetylcholine is a dominant neurotransmitter in the neuromuscular junction. Neurotransmitters are chemical messengers that transmit a message from a nerve cell across the synapse to a target cell. The target can be another nerve cell, or a muscle cell, or a gland cell. They are chemicals made by the nerve cell specifically to transmit the message. We will discuss later how these tiny chemical substances are formed and released. But before we get there, we will discuss first how these interact with the motor end plate. When a nerve impulse reaches the neuromuscular junction, about 125 vesicles of acetylcholine are released from the terminals into the synaptic space. They function to receive the acetylcholine that were previously released from the presynaptic membrane. Alright, so diving in deeper are the features of an acetylcholine receptor. On the inside surface of a neural membrane are linear dense bars, and to each side of each dense bar are protein particles that penetrate the neural membrane, and these are the voltage-gated calcium channels. So dense bars are areas where acetylcholine vesicles empty into synaptic throat, and active zones lie in close vicinity to dense bars, specialized release of acetylcholine vesicles. We have the basal lamina, which contains acetylcholinesterase, an enzyme which destroys the acetylcholine. So we will know about this enzyme later on. Calcium ion channels are located in the neural membrane near the mouths of the subneural clefts. And the synapsin, which I just mentioned a while ago, would anchor the acetylcholine vesicles to the cytoskeleton of presynaptic terminals. Alright, this is actually not found in the image, but it's there. So going back to this slide, when an action potential spreads over the terminal, the voltage-gated calcium channels open and allow calcium ions to diffuse from the synaptic space to the interior of the nerve terminal. The calcium ions, in turn, are believed to activate calcium calmodulin-dependent protein kinase, which, in turn, phosphorylates synapsin proteins that anchor the acetylcholine vesicles to the cytoskeleton of the presynaptic terminal. This process frees the acetylcholine vesicles from the cytoskeleton and allows them to move to the active zone of the presynaptic neural membrane adjacent to the dense bars. The vesicles then dock at the release sites, fuse with the neural membrane, and empty their acetylcholine into the synaptic spaces by the process of exocytosis. Alright, so we'll tackle on the acetylcholine when it opens ion channels on postsynaptic membranes. This is an example of an acetylcholine gated channel, and in picture A it shows you a closed state. Note that the negative charges at the channel mouth prevents passage of negative ions such as chloride ions. As acetylcholine has become attached and a conformational change has occurred, this will open the channel, allowing sodium ions to enter the muscle fiber and excite contraction. The principal effect of opening the acetylcholine gated channel is to allow large numbers of sodium ions to pour to the inside of the fiber, carrying with them large numbers of positive charges. And this action creates a local positive potential change inside the muscle fiber membrane called the EPP, or end plate potential. In turn, this end plate potential initiates an action potential that spreads along the muscle membrane, and this causes muscle contraction. Alright, so these are the acetylcholine channels. They are also known as acetylcholine gated channels. They are composed of large protein complex of 5 protein subunits that penetrate all the way through the membrane, lying side by side in a circle, thus forming a tubular channel. The diameters are large enough to allow important positive ions like sodium, chloride, and calcium to move easily through the opening. Strong negative charges on the mouth of the channel repel negative ions like chloride. So, Doc, why would there be more sodium ions that flow through acetylcholine channels than potassium? So here is your answer. There are two positive ions in great concentration. Your sodium is in the ECF and your potassium is in the ICF. The very negative potential on the inside of the muscle membrane pulls the positive sodium ions to the inside of the muscle fiber, while potassium is attracted to the negative ions at the mouth of the acetylcholine channels, which prevents the potassium efflux when they attempt to pass outwards. And this creates a local potential inside a fiber called an end plate potential, or your EPP. So the figure here, this is actually an end plate potential in millivolts. This shows you the principle of an end plate potential initiating the action potential. This figure shows 3 separate end plate potentials. End plate potentials A and C are actually too weak to elicit an action potential, but they do produce a weak local end plate voltage changes as recorded in this figure. By contrast, end plate potential B is much stronger and causes enough sodium channels to open so that the self-regenerative effect of more and more sodium ions flowing to the interior of the fiber initiates an action potential. So we go to the overview of neuromuscular transmission.
The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
Alright, so just a brief overview of what we just saw. So secretion of acetylcholine by nerve terminal is brought about by the action potential arriving in the presynaptic motor nerve or neuron axon terminal. Depolarization of the plasma membrane axon terminal. Voltage-gated calcium channels open, increasing the permeability to calcium in the axon terminal, which causes calcium ions to exert attractive influence on acetylcholine vesicles. Acetylcholine vesicles then drawn close to the neural membrane adjacent to the dense bars, if you remember. And acetylcholine vesicles release acetylcholine by exocytosis. Acetylcholine then diffuses to the post-junctional membrane or the muscle cell membrane, to which acetylcholine combines with specific receptors at the post-junctional. Acetylcholine channels open and thereby increasing the permeability of post-junctional membrane to sodium and potassium, and an end plate potential or an EPP is created. Depolarization of the post-junctional plasma membrane occurs, and depolarization wave travels in both directions toward the muscle fiber ends, creating an action potential and thereby creating movement. Yay! Congratulations! Of course, all of these things happen in a split second. You thought we were done? Nope. I have two more topics or three more topics to discuss, and this is one. We will discuss how these tiny chemical substances are formed and released. Ring a bell? Ay lagi sige og doka.
Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Alright, so we'll discuss about the stages of acetylcholine formation and release and also their retrieval. So first up, we'll discuss about the formation. So small vesicles, about 40 nanometers in size, are formed by the Golgi apparatus in the cell body of a motor neuron in the spinal cord. And these vesicles are then transported by axoplasm that streams through the core of the axons from the central cell of the body in the spinal cord, all the way to the neuromuscular junction at the tip of the peripheral nerve fibers. And about 300,000 of these small vesicles collect in the nerve terminals of a single skeletal muscle and plate. Acetylcholine is then synthesized in the cytosol of the nerve fiber terminals, but is immediately transported through the membranes of the vesicles to their interior, where it is stored in highly concentrated form, about 10,000 molecules of acetylcholine in each vesicle. Once again, when an action potential arrives at the nerve terminal, it opens many calcium channels in the membrane of the nerve terminal because this terminal has the abundance of voltage-gated calcium channels. As a result, the calcium ion concentration inside the terminal membrane increases about 100-fold, which in turn increases the rate of fusion of acetylcholine vesicles with the terminal membranes about 10,000-fold. And this fusion makes many of the vesicles rupture allowing exocytosis of acetylcholine into the synaptic space. About 125 vesicles usually rupture with each action potential. Remember that. Then after a few milliseconds, the acetylcholine is split by acetylcholinesterase into acetate ion and choline. And the choline is reabsorbed actively into the neural terminal to be reused to form any acetylcholine or a new acetylcholine. So this sequence of event occurs within a period of 5 to 10 milliseconds. So this is a quantum release of acetylcholine. So acetylcholine is released in packets or intermittently and not continuously, and each packet corresponds to the release of one synaptic vesicle. The number of vesicles available in the nerve ending is sufficient to allow transmission of only a few thousand nerve-to-muscle impulses. Therefore, for continued function of the neuromuscular junction, new vesicles need to be reformed rapidly. Within a few seconds after each action potential is over, coated pits appear in the terminal nerve membrane caused by contractile proteins at the nerve ending, especially the protein clathrin, which is attached to the membrane in areas of the original vesicles. Within about 20 seconds, the protein contracts and causes the pit to break away to the interior of the membrane, thus forming new vesicles. Within another few seconds, acetylcholine is transported to the interior of these vesicles, and they are then ready for a new cycle of acetylcholine release. Alright, so there are drugs that enhance or block transmission at the neuromuscular junction. So this is just short. So there are three categories of drugs that enhance or block the transmission in the neuromuscular junction. The first one we have are the drugs that stimulate the muscle fiber by acetylcholine-like action. So several compounds including methacholine, carbachol, and nicotine have nearly the same effect on the muscle fiber as does acetylcholine. So for short, these drugs mimic acetylcholine function. Just a difference between these drugs and acetylcholine is that the drugs are not destroyed by acetylcholinesterase or are destroyed so slowly that their actions often persist for many minutes to several hours. The drugs work by causing localized areas of depolarization of the muscle fiber membrane at the motor end plate where the acetylcholine receptors are located. Then every time the muscle fiber recovers from a previous contraction, these depolarized areas, by virtue of leaking ions, initiate a new action potential, thereby causing a state of muscle spasm. The second one is that drugs that stimulate the neuromuscular junction by inactivating acetylcholinesterase. So three particularly well-known drugs, the neostigmine, physostigmine, and diisopropyl fluorophosphate. They inactivate acetylcholinesterase in the synapses so that it no longer hydrolyzes acetylcholine. Therefore, with each successive nerve impulse, additional acetylcholine accumulates and stimulates the muscle fibers repetitively. And this activity causes muscle spasm when even a small nerve impulse reaches the muscle. Unfortunately, it can also cause death as a result of laryngeal spasm, which smothers the person. Neostigmine and physostigmine combine with acetylcholinesterase to inactivate the acetylcholinesterase for up to several hours, and after which these drugs are displaced from the acetylcholinesterase so that the esterase once again becomes active. Conversely, diisopropyl fluorophosphate, which is a powerful nerve gas poison, inactivates acetylcholinesterase. Last but not the least are a group of drugs known as curariform drugs. So these drugs are the ones that block transmission at the neuromuscular junction. So these drugs can actually prevent passage of impulses from the nerve endings into the muscle. So for an instance, D-tubocurarin blocks the action of acetylcholine on the muscle fiber acetylcholine receptors. and thus preventing sufficient increase in permeability of the muscle membrane channels to initiate an action potential. Remember this picture? I explained it a few slides ago, just so you know. So please go back on that part if you fell asleep. So the weakness of the end plate potential, I have actually not mentioned this a while ago, but the weakness of the end plate potential at point A was caused by a poisoning of the muscle fiber with curare, a drug that blocks the gating action of the acetylcholine on the acetylcholine channels by competing for the acetylcholine receptor sites. And the weakness of the end plate potential at point C resulted from the effect of a botulinum toxin, a bacterial poison. Right, so these are the different agents for affecting neuromuscular transmissions. So please just read on the examples and then know the actions and the effect on the neuromuscular transmissions. Last but not the least, these are the diseases associated with pathological conditions at the neuromuscular junction. So there are just there are two diseases associated with the neuromuscular junction. We have myasthenia gravis, abbreviated as MG, and Lambert-Eaton syndrome, which is abbreviated as LES or LES. For MG, you need to remember that this targets the postsynaptic terminals, and acetylcholine is abundant, but the receptors are not working. The more you stimulate the receptors, the less likely it will function, which explains why the person will be normal in the morning, but they tend to become very weak in the afternoon. LES or LES, on the other hand, affects the presynaptic terminals and is caused by the presence of circulating antibodies against voltage-gated calcium channels in nerve terminals, and therefore few release of acetylcholine from the synaptic vesicles happen. But with continuous stimulation, the physical aspect will actually improve the body weakness since ACH gets accumulated in the long run. So that's it. So thank you for watching this video. If you have any comments or concerns or questions, please don't hesitate to comment down below. By the way, while my videos are uploaded in YouTube, they are actually unlisted, which means you are the only ones able to access them. Once again, thank you and have a great day ahead. Stay safe. I never get tired of this.